So till now we have studied about the microscope, that by using the optical light or by using simple light, we will illuminate our substance, and depending upon that we study the particular substance. But electron microscopy is a different kind of microscopy. Electron microscopes are the instruments that uses a beam of electron, or I should say highly energetic electron, to examine the objects in a very fine scale. That means when we want to identify our compound or any other matter or any particular substance, smallest atom also, at nano level or below that or just near to nano level, we have to use electron microscopes. So the technique which uses electron microscope to understand or to identify the compound is known as electron microscopy. Okay? Here, electron microscopy basically involves which things? So, the examination information, or I should say, the substance information which can be provided to us by the electron microscopes are topography, morphology, composition and crystallographic information. So, by doing the study of any substance using electron microscope, we will get an idea about the surface characteristic of the substance, which is the topography, the morphology of the substance, which is basically the structure of the compound, the composition of the substance, which is give us an idea about which kind of atoms or which kind of molecules are actually present in the compound. And the last one, it gives us an idea about the crystallographic information of a particular substance. So we get overall study of a substance using electron microscopes. Okay? So these are the informations which we can obtain using electron microscopy. Next one is the types. The types of electron microscopes are of two. Number 1, SEM, and number 2, TEM. Let's see their full forms. So, SEM is basically scanning electron microscope, while TEM is transmission electron microscope. SEM is used to visualize the surface of the objects, while TEM is used for the study of the internal structure of the object. So I should say that SEM is a surface identification, while TEM is an internal identification of a particular compound. Okay, so let's begin with that. The first one which we'll have here is SEM. SEM, the full form of SEM is scanning electron microscope or microscopy. In the image you can see the instrumental setup for scanning electron microscope. The room should be soundproof. The internal environment of the instrument is totally under vacuum. So the room has to be fully and 24/7 air conditioned in order to maintain the temperature, in order to maintain the vacuum, in order to maintain the conditions of that particular instrument, right? Each and everything, for an example, the pumps, that means the vacuum pumps, the data system, the computers should be supercomputers which can detect a very high definition images, that has to be there. Along with that, the proper sample preparation kit has to be there. So all these are the requirements when we deal with the scanning electron microscopy. Now, last but not the least, a very and a knowledgeable, properly trained person should be there to handle scanning electron microscope or for the identification of the compound through scanning electron microscopy. Okay? Now, Which are the things that are involved? So an electron microscope that produces the images of the sample by scanning over it with a focused beam of electrons is known as scanning electron microscopy. Okay? Now, the incident electrons interact with the electrons in the sample, producing various signals, or produces secondary electrons, right? So the electrons actually bombarded on the sample, and after that they produces the secondary electrons, and afterwards we get them, we catch them, and we identify the particular substance. Okay? So they are detected and they contain information about the sample surface topography as well as surface composition can also be detected. How it's possible? So when SEM gets connected with EDXRF, at that time SEM determines the surface topography, while EDXRF determines the composition of the particular surface, that which metal actually presents in which amount on the surface of that particular substance. Okay? So these are the hyphenated methods. What we understood by that? Two different methods combines together to produce one whole technique, and it is known as highly hyphenated methods. Okay? Now next one, let's observe the principle. Here, the first thing is the electron gun. On top of the instrumentation, you can see electron gun is there. After that, it produces the electron beam. Now this electron beam is in each and every direction. So we use anode in order to get it directed in a particular direction, or to get concentrated electron beam. After that, through magnetic lens, it is focused on the sample. Okay. Afterwards, it will be passed through the scanning coils. At last, totally they are focused on our sample. At last we have a sample stage which contains the specimen and the electrons are focused on it. Afterwards, when the electrons actually react with the sample produces other electrons. They are known as backscattered electrons or secondary electrons, right? They get captured using scanning electron coils or scanning coils using the backscattered electron detector and other secondary electron detectors, and they move to the TD scanner, or I should say the data system, and afterwards the graph can be produced. Okay? So here, this is the operation procedure, right? Now, which are the things that contain in the instrument of it? So first one is electron gun. The electron gun is actually a filament which produces a highly or high speed and highly energetic electrons, right? So after electron gun, it has condenser lenses. What is the work of this condenser lenses? They focus the beam on a particular direction. Okay? Afterwards, objective aperture. Objective aperture is an aperture which focuses it on a total specimen or on our specimen. Afterwards, we have scan coils. Two solenoids can be used because we have to detect secondary electrons also and we have to detect the backscattered electrons also. So to detect them, we use this kind of solenoids. Afterwards, we have a chamber which is a specimen stage or specimen chamber. Okay? And last one, computer hardware. That means it goes to detectors, and after that, computer hardware and computer software, which is used for the interpretation of the results. Okay? Now, here you can see the internal picture of the chamber, of specimen chamber. Here you can see three different detectors are there. Number one, EDS. Number two, secondary. Number three, backscattered electron detectors. Right? Three different kinds of electrons are produced, and depending upon that, we have to use three different detectors at different angles in order to capture the electrons. Right? This is the base of scanning electron microscopy, and we have to understand it properly in order to get the actual image of the surface topography. Now let's see the applications of scanning electron microscope. So SEM has very high practical, industrial and technological applications such as semiconductor inspection, production line of minuscule products, as well as assembly of microchips for the computers. We all know that we have now a trend of nanochips. So nanochips have a structure at a nano level. Now if you want to analyze that how the particular socket is joined with each other, at that time you have to take the pictures. The pictures can be possible using SEM, right? So this is the best application of scanning electron microscope. Likewise, SEMs can be essential in research tool. For example, to study scientific principles for biology, for pharmaceuticals, even they are widely used in gemology, medical and forensic sciences, and even metallurgical operations also. It includes the study of gems. Okay? And diamonds are also identified using scanning electron microscopes. In addition to the topographical, morphological and compositional informations, SEM can detect and analyze surface fractures, which provides us the best examples in microstructures, examines surface contaminations, reveals spatial information or variations in chemical compositions. Even they can provide qualitative and quantitative chemical analysis of any crystalline structures also. So in that manner we can say that SEM has a very wide application, or they have a very good scope in future.